Hi, everybody. It's a pleasure to. It's a pleasure to be once again here for another installment of the Immuno webinars. Today we are going to have a talk by Dr. Leonardo Travassos, who is a graduate. Let's get my note here. He's a graduate in biological science from the State University of Rio de Janeiro, got his master's also from the State University. And he's at the University of Rio de Janeiro. He did his postdoc training period at the Department of Immunology at the University of Toronto, and since 2010, he's part of the Biophysics Institute at the Federal University of Rio de Janeiro, where he's the head of the laboratory in immunoreceptors and signaling at the immunobiology program. Dr. Leonardo is also holds a fellowship from the state funding agency as cientista do nosso estado, and was an affiliate member of the Brazilian Academy of Sciences. He works mainly with autophagy, focusing on the mechanisms related to the response to intracellular pathogens. He published more than 45 articles in journals, and which had received so far more than 10,000 citations for an age index of 25. He also supervised lots of students, 6 PhD students, 7 masters, 12 scientific initiation. He's active both at CNPq level, at the state funding agency, and at the Brazilian Society for Immunology. So it's really a great pleasure today to receive Dr. Leonardo today, and to listen to him talk. So Leonardo, please. Microphone. Então, vai rolando aí enquanto eu abro aqui. Ó.
Só falar normalmente?
Só falar normalmente.
Ah, tá.
Falando aí, sem DDA. Né? Broma aí. Não, é só porque eu não consigo usar o computador. Vocês só gastar programa.
Porra. 97. Não, mas a apresentação não vai ser nisso. Isso é uma brincadeira. Ele passa? Mas não está passando.
Esse que vira o mouse. Esse funciona com o mouse. Ele não pode funcionar com o poder. É assim que fica.
Ok, ok.
Você passa o slide para frente, é só você voltar. Esse aqui e aí, se você aperta seguro, vai aparecer, aí você pode mexer no mouse.
Esse aqui aperta e segura.
Ele só vai aparecer na tela.
Tem que ligar ali, né?
Não, já tá.
Tem que fazer uma pós-graduaçãozinha. So, okay, thank you. I would like to start by thanking the organizers for inviting me. So I was finally invited by Christian. I'm kidding. She's trying to invite me, and I'm always posting. I would like to thank for the presence of my dear friends, people on Zoom. So I'm not going to the first PowerPoint background I used when I started to use computers. So don't worry. Today we are going to talk about on YouTube. I'll give you an overview about
Ô, Léo,
autophagy. Yes.
Bruno, desculpe aí, o som, não sei se a internet aí não está funcionando direito, porque está dando um sinalzinho de vocês bem vermelhinho, assim, bem ruinzinho. E o som não está chegando aqui.
Você pode ficar daqui do lado.
Os dois estão ligados. Só que tá.
Para os dois ligados.
Okay, so let's try. So because we have many students here, I'm going to give you an overview on autophagy, some historical perspective. Here are some of the topics we are going to approach today, not necessarily in this order. So autophagy is this highly conserved degradative process in which we have cytosolic material delivered for degradation in the lysosome. Okay, so it's a highly dynamic process, as I mentioned before, highly conserved, and it comes in three versions: macro autophagy, micro autophagy, and chaperone-mediated autophagy. So for the sake of this presentation, we are going to focus on macro autophagy, in which we have the formation of this double layered membrane called autophagosome, which is the hallmark of the autophagic process. So upon selection of the cargo, we're going to talk about cargo selection throughout the presentation. Upon completion of this autophagosome, it's going to fuse with the lysosome, and the material, the degradation product, can be reused for energy purposes. So autophagy is this very complex process. Here I have a simplified schematic view of the autophagic pathway. We have some protein complexes that will act in a very hierarchical manner, leading to the formation of the autophagosome, right? So we have this first protein complex, that the ULK complex, that's gonna integrate signals from very energy sensing pathways, such as mTOR and AMPK, allowing to the initiation of the autophagic process. Okay, then we have the second protein complex, the PI3 kinase complex that's gonna generate inositols that are necessary for the nucleation of the pre-autophagosomal membrane. And then we have this these two complexes, the ATG5-12-16 complex and the ATG8 complex, that will act in concert to expand the autophagosomal membrane, right? And this will lead to the lipidation of this protein, LC3, that upon lipidation will be specific for the membrane of the autophagosome and is so far the best marker for tracking autophagy, right? So I'm going to present some historical view of the field. This is a review from Daniel Klionski, someone very important for the field, and I'm going to discuss very quickly some of the main findings of this timeline. So at the beginning of this century, someone told Daniel Klionski that autophagy would be the new apoptosis, right? So this sentence seemed very bold and audacious, but eventually it proved to be true. After 4 to 5 years, autophagy emerged as this very important homeostatic process that has its fingerprints in fields such as cancer biology, immunity, lifespan, neurodegeneration, and many others, right? So it all begins in 1955 with Christian de Duve, which described the lysosomes. He received he was awarded with the Nobel Prize in '74. But he made the first description, morphological descriptions of the autophagosome. And he also coined other terms that we used until now, like exocytosis and endocytosis. They were all coined by Christian de Duve. But from its first description until the 80s, autophagy was merely a morphological process, okay? And much of the work was centered on the final steps of the autophagic process, meaning the degradation process, okay? And for these reasons, autophagy was considered a non-selective degradation process. And everything starts to change in 1993, when Osumi's lab, in a screen for in East, in a screen for autophagy mutants, he identified these mutants that we can see here, that the vacuoles from these mutants are empty, okay? So there was no delivery of material for degradation in the autophagosomes. Later, the group from Majer, they found that PI3 kinases inhibitors such as wortmannin and LY, they blocked the formation of autophagosomes. And interestingly, these tools are used so far, right? So until now, we have many papers using these drugs to demonstrate that this, whatever process is dependent on autophagy. Right. Then, in '97, again, Osumi's lab cloned the first autophagy gene in East, and Mizushima identified the first ATG autophagy gene in mammals. Okay, so far we had a lot on autophagy in East, but not so much in mammals. This paper demonstrates that ATG5 that is also present in East. It participates in the autophagic process in mammals, always in complex with ATG12. Then Beth Levine in '99 demonstrated for the first time a connection between autophagy and cancer. So using this this breast cancer cell line that does not express, naturally does not express Bactin-1. Her group demonstrated that upon injection of this cell line in mice, we have the formation of large tumors. But when this same cell line was complemented with Bactin-1, the size of the tumors was way smaller, and they also demonstrate that many breast cancer cell lines do not express Bectin, right? So in 2000, Yoshimori and colleagues, they identified LC3 as a marker for the autophagosomal membrane. And this was really important because it allowed many groups around the world to start to work with autophagy. David Rubinstein's lab in 2002 demonstrated a role for autophagy in the degradation of toxic protein aggregates that are characteristic for Parkinson's disease, Huntington's disease, for example. So, and again, Beth Levine and another group, they demonstrate that autophagy was important for the extension of lifespan, and they use this C. elegans model. So autophagy deficient C. elegans has a shorter lifespan. In 2004, we had this interesting paper demonstrating that autophagy is important for neonatal mice. So after delivery, mice, they do not start to suck immediately, so there is a lag. And normally, these animals, they have to do a lot of autophagy to cope with this, let's say, 24 hours without nutrition, right? So autophagy deficient mice, they will die within 24 hours because of a decrease in levels of total, essential, and branched chain amino acids, and they show the same for glucose and other markers. Okay, so autophagy is very important in the neonatal period for mice. We'll be back to this timeline when we start to talk about autophagy and bacteria, right? So very quickly, I would like to comment on autophagy and diseases. We have strong connections between autophagy and different diseases. I will comment very quickly on neurodegeneration and cancer. So it's pretty clear that autophagy is important for the homeostasis of the nervous system. And it's important in its different steps. So here we can see that the effects in the initiation, in the induction step is really important. We have mutations or SNPs correlated with diseases such as Laffer's disease, Huntington, and Parkinson. We have defects in the cargo selection also related to neurodegeneration. So defects in mitophagy or defects in this protein P62 which we'll talk a lot throughout the infection, have also been related to neurodegeneration, and also defects in the final step of the autophagic process, meaning the fusion of lysosomes and autophagosomes have also been related to, linked to neurodegeneration. But probably the best example of the importance of autophagy and in diseases comes from cancer. The currently accepted idea is that autophagy, which is important for the maintenance of genomic stability. So autophagy is anti-tumorigenic under steady state conditions, right? This is currently accepted. However, upon neoplastic transformation, autophagy clearly plays a pro-tumorigenic role, okay? And this creates what people in the field of cancer and autophagy, which is not my thing, they call it the autophagy paradox, right? So here we have some examples in which autophagy can be either protective or protomorigenic before the installation of transformation, okay? So it is kind of still debatable, but clearly upon transformation is initiated, we have a protomorigenic role for autophagy in very different models, right? So many attempts to find drugs that can block autophagy and be used as treatment for different types of cancers. Most of them focus on the final step of the degradation process, okay? So they try to block lysosomal acidification, for example, or the fusion of the autophagosome with the lysosome. Okay, so back to the timeline of Daniel Klionski. In 2004, several groups demonstrated autophagy as a factor of the innate immune system, right? So, and how I came across the idea of autophagy and bacteria. So I had this background. I was returning from Sandwich in Paris, in which I worked in the group of Dana Philpott, and they had just demonstrated that not one and not two are sensors for MDP and peptidoglycan. So meaning not one, not two, they sense the presence of peptidoglycan in the cytosol of cells that are infected, okay? And interestingly, during this study, they found that TLR2, that peptidoglycan was not an activator of TLR2. So let's context it in a
Léo, querido, seu som sumiu.
Não, também não estou ouvindo, não.
Oi, can you hear me?
Agora sim, agora sim.
Voltou.
Okay. So the TLR field started in, let's say, 97, 98, 2003, still in expansion, but we still had problems with the use of contaminated products from Sigma or other companies. So LPS was not pure, peptidoglycan was not pure, lipoprotein was not pure. And we had many papers demonstrating that TLR2 was a sensor for peptidoglycan, okay? Just as an example, we had a paper on Nature demonstrating that TLR2 was a receptor for LPS, right? Which we now know it's a mistake due to the use of contaminated LPS. So the LPS they used was, I think they bought from Sigma, and it was contaminated with lipoproteins, okay? So Dana's group had this observation that pure peptidoglycan could not activate TLR2. So we decided to investigate that in more detail. So we partnered with an expert in peptidoglycan, Ivo Boneca, from Portugal. And so we purified peptidoglycan from different gram-positive and gram-negative bacteria. Here we have, I'm showing only 4 gram-positive, but we have exactly the same 4 gram-negative bacteria. And these numbers here, they mean the purification step of the protocol, right? So first, second, and third purification step of the protocol, and we see that the more we purify the peptidoglycan, the more we have not too dependent activation of NF-kappa B, but the more we purify the peptidoglycan, the less we have TLR2 dependent activation of NF-kappa B, right? So with that paper, we demonstrated that TLR2 was not a sensor for peptidoglycan, which make not one or two the only known sensors for bacterial peptidoglycan, okay? So just as a curiosity, I went to a meeting in São Paulo, and I was presenting a poster. That was 2004. And Douglas Goldenbach came to the poster, and he was saying, how you guys have the guts to show that, to say that TLR2 does not sense peptidoglycan? My paper has 500 something citations. And I said, well, you know, listen. And then Douglas, and he said, well, I know, I'm just teasing you. I know the PG we used was contaminated. So it was really a problem. And we had to, I mean, many situations because of this paper. We had a paper after that, not us, but another group from Roman Ziziarski, a paper on infection immunity, just fighting this one and saying, I mean, refuting each of the experiments we did. But I mean, in the end, this idea prevailed, and TLR2 does not sense peptidoglycan. Okay, so I was back in Brazil, and I was participating in these seminars, topics in innate immunity, something like that, organized by Marcelo. And we had lots of papers on TLR, on new TLRs being presented. So nice papers on Nature, Cell, Science, and other high-profile journals with new TLRs, new protein adapters. But eventually, these papers, they were all the same in terms of methodology, and we discussed, well, let's try different approaches, different papers. I was the next one to present. So here, just we had many colleagues, many people you know, Renata, Diego, it's in the US, Rodrigo Tinoco, Heitor, Barbara, which is in Canada now, Lilian. Maybe André was, I don't know if you know. No, not here yet. Okay. So, but this paper, which was the one, my first contact with autophagy was through this paper on Science. They showed that HeLa cells infected with group A Streptococcus, we can see the bacteria in red, perfectly surrounded by autophagosomes in green, right? And this really caught my attention, and it's been like this until today. They demonstrate that autophagy deficient cells, they have a defect in controlling the infection, right? So really, really nice result. A few months later, we had more or less the same, but with macrophages infected with BCG. And epithelial cells, another one on Science, with epithelial cells infected with Shigella, right? So when I saw this picture, the first thing that came to my mind was where the specificity comes from, how the autophagy machinery can perfectly segregate bacteria from the rest of the cytoplasm, right? And because of this background in peptidoglycan recognition and ligand identification, I thought that NOD1 and NOD2 could participate. So we knew that NOD1 and NOD2, they sense peptidoglycan. They recruit RIP2 to activate NF-kappa B and the production of pro-inflammatory cytokines. And I thought that they could also, by sensing PG, trigger autophagy. And this, again, just a quick joke. This was the slide that I presented when I arrived in Toronto with my idea, which is this very slide here with this ugly, again, PowerPoint background. I'm not very good with PowerPoint design and stuff. So those were my arguments to say, well, I think we need to look into this thing. So after those papers, those 3 papers that I just showed you, we had people from the innate immunity field jumping into autophagy, and we quickly had this paper on immunity and this one on EMBO journal, demonstrating that TLR ligands are able to upregulate autophagy. So general upregulation of autophagy, but in a TLR dependent manner. But again, no clue about the specificity. And then we started to look into a possible role for NOD1 and NOD2 in triggering autophagy. So we did experiments in which we simulated cells with NOD1 and NOD2 ligands, and we have an upregulation of the autophagic process. In vivo, we had the same. When we infect cells with Shigella, we had this the activation of bacteria targeted autophagy, which is absent in ATG5. And NOD1 deficient cells, we have the same results for NOD2. NOD1 and NOD2 deficient cells, they do not trigger bacteria targeted autophagy as efficiently as the wild type counterparts. Okay, and this was not dependent on RIP2 and not dependent on NF-kappa B activation. And things were running really well, but then in 2007, we had this first paper followed by many others demonstrating a single nucleotide polymorphism in ATG16 as the second most important genetic risk factor for Crohn's disease. The first was NOD2. So we couldn't, we could not just ignore this information. We had to somehow approach these findings, and eventually we demonstrate that NOD1 and NOD2, they are important to recruit ATG16 to the bacterial entry site, okay? Using different ATG16 and different NOD2 mutants, we found that wild type NOD2 recruits ATG16 to the bacterial entry site. So this is 5 to 10 minutes infection. We have the colocalization of NOD1 and ATG16, or NOD2 and ATG16. And interestingly, the NOD2 mutation, the most common NOD2 mutation that people call this NOD2 frame shift mutation, this mutant was not able to recruit ATG16 to the bacterial entry site. Okay, so we finally did experiments with this with macrophages from this NOD2 frame shift knocking mice that we got from Gabriel Nunes, and we demonstrate that these mice, they have defect in bacteria targeted autophagy. Okay, so and eventually we came up with this model. So NOD2 is important for triggering inflammation. NOD1 and NOD2, but also to trigger bacterial targeted autophagy. So this paper came out end of 2009, very end of 2009. The very beginning of 2010, we had a paper on Nature. There was another paper on Nature Medicine showing pretty much the same. Right, so we know now that CLR, innate immunity receptors, they participate in providing specificity to the system, but there are other proteins as well. So more or less by the same time, many groups were looking for proteins that could participate in substrates in cargo selection, so for the delivery for lysosomal degradation, right? So we have this explosion in the research for what people call SARS, for selective autophagy receptors, okay, that participate in many subtypes of autophagy, such as mitophagy, pexophagy, ER-phagy, lysophagy, and so on. Okay, so the most studied one is P62. P62 is this ubiquitin binding protein that binds ubiquitin, but at the same time, because of due to the presence of this LIR domain, so LIR stands for LC3 interacting region. So P62 can bind to ubiquitin and LC3 at the same time. So it can detect the ubiquitination of different substrates and deliver these ubiquitinated substrates for degradation in the autophagosome, okay? And quanto tempo tem? Okay, so we show that out of this protein, P62 is important for the removal of protein aggregates, this protein aggregates that we described, Alice, a while ago. So autophagy deficient cells, they harbor way higher numbers of these protein aggregates. This is dependent on NRF2, heme oxygenase in the presence or in the release of iron from heme. Okay, I forgot to say. It's really important. So these aggregates, they are generated upon the stimulation of macrophages with heme. Okay. Right. So let's speed up a bit. And this is also through the formation of this protein aggregates is also true for TLR ligands. So many TLR ligands induce the formation of this protein aggregates in a TLR4 and TLR2 dependent manner for LPS and PAN-CIS. Okay, so back to xenophagy. We had many groups studying, describing proteins involved in cargo selection. And more or less by the same time, we had the first evidence that bacteria present in the cytosol are ubiquitinated. Okay, so they are decorated with ubiquitin. Okay, so bacteria are present in the cytosol, they get ubiquitinated, but to get ubiquitinated, we need this ubiquitin ligases such as SMURF1, Parkin, and others that will add the ubiquitin to the bacterial surface. Okay, so we know that bacteria trigger xenophagy. Pattern recognition receptors, they mediate xenophagy. Bacteria are ubiquitinated. We need ubiquitin ligases, but we don't know what are the signals that control the ubiquitination of bacteria, right? So we thought that maybe the same complex that integrates signals from mTOR and AMPK could to trigger autophagy could also be the responsible for the control of the ubiquitination step. So we started some experiments. We demonstrate that the infection of MAPS with Listeria induces a state of amino acid starvation, which is something that is sensed by ULK, right? So by looking for the delocalization of LAMP1 and mTOR, we were able to demonstrate that bacterial infection triggers amino acid starvation. And then let's jump ahead a bit. We thought that maybe ULK1 would be, which senses amino acid starvation, could be responsible for ubiquitination as well. So we did some experiments to demonstrate that infection with Listeria triggers xenophagy, triggers bacterial ubiquitination. It depends on the escape of the bacteria to the cytosol, and also recruits P62, right? So using ULK1 deficient cells, we demonstrate that these ULK1 deficient cells, they have defect in the recruitment of ubiquitin and P62 to the bacterial surface. And this has an important impact in the replication of bacteria. So ULK1 deficient cells, they have a defect. I mean, they control the infection less efficiently than the wild type. Interestingly, this is true only for wild type bacteria, bacteria that are not able to do cell-to-cell spreading. So this is a Listeria mutant that does not make cell-to-cell spreading. The ULK1 is not important for ubiquitination or recruitment of P62. We are still trying to understand what's happening here. But in terms of mechanism, we know from the literature that ULK1 controls the phosphorylation of P62 in this residue S409, right? So this phosphorylation is important for the increase in the affinity of P62 for ubiquitin, okay? So this group demonstrated that in a context of proteasome inhibition, this phosphorylation that is ULK dependent is important for the recruitment of P62 to ubiquitinated protein aggregates. And we saw that it could maybe be true also for bacterial infections. Okay, so we use this P62 deficient MAFs that were retroviolet transduced with this mutant, phosphorylation mutant, S409A. So we demonstrate that these cells harboring these mutants, so a non-phosphorylatable P62 at this position, S409, has an important defect in the recruitment of P62, leads to a defect in the recruitment of LC3, and a defect in the control of bacterial replication, okay? So I'm going to change a bit the subject. It's still autophagy and bacterial infections. We've been interested in understanding how lipophagy is implicated in the response to cytosolic bacteria, right? So we know that metabolic adaptation of host cells is a key determinant to the fate of infection. Pathogens target host metabolism to create replicative niches, and lipid metabolism is heavily affected by infection. So pathogens, they modulate host plasma membrane for their bacterial entry. They recruit specific lipids to keep the integrity of their vacuoles. And they also use host lipid droplets for the regulation of immune responses and for energy production. So lipid droplets are these very dynamic and highly conserved organelles that are present in mammals, in different cell types. Here is an adipocyte, macrophage, in plants, in C. elegans. Even bacteria have this lipid droplet-like structures. So in terms of function, they are for sure not inert fat particles. They they function as energy storages. They prevent lipotoxicity. They buffer oxidative stress. They are platforms for lipid-derived signaling and innate immune responses. And they have been implicated in different diseases such as obesity, arteriosclerosis, infection, cancer. So for those interested in the function and structure, I really suggest you guys to read this review from Patricia's group. They really nailed the function of these organelles in this review. So we know, due to the work of Patricia, Cristiani, and many others, we know a lot about the stimuli that leads to the biogenesis of lipid droplets and their role in infection, but not too much on how important, or if it is important, to remove these organelles after their biogenesis. So, and because we know that autophagy is important for the degradation of lipid droplets. So in this paper in 2009, the group from Ana Maria Cuervo demonstrates that autophagy is key for the removal of lipid droplets. We thought that maybe it would be interesting to understand if removing these organelles after or during infection would be important for the fate of infection. So very quickly, what we've been doing in the last months. So we infected immortalized bone marrow-derived macrophages with listeria, stained with lipid tox to stain lipid droplets. And we quantified these lipid droplets, and we can see that they have there's a peak of accumulation of lipid droplets at 8 hours. We tested different MOIs, different time points. But then in all MOIs, we have this decrease in the number of lipid droplets by 24 hours. So basically, the numbers, they return to basal levels, okay? So we had the same using another macrophage cell line, the same kinetics, a peak peaking at 8 hours, then returning to normal to basal levels in 24 hours. We saw that the formation of these organelles, their biogenesis, their accumulation, depends on the escape of the bacteria to the cytosol. Okay, so mutants that do not escape to the cytosol do not induce the accumulation of lipid droplets. And then we started to see if we had the induction of lipophagy. Okay, so I apologize for this things appearing here. This is not appearing in the computer screen. But what we see here are bone marrow-derived macrophages, immortalized ones, infected with listeria, and we can see the colocalization of lipid droplets in green with LC3 in red, in wild type, but not in ULK1 deficient cells. Right? So we quantified the colocalization using plugin on ImageJ, and we can see this a significant difference in the colocalization of lipid droplets with autophagy markers. So we saw for LC3, and we saw the same for LAMP1. Okay, so clearly the infection triggers lipophagy. Then we infected wild type and ULKO deficient macrophages for different time points with listeria again, and we can see that while at 24 hours, while the wild type macrophages, the infected ones, the number of lipid droplets returned to basal levels, the same does not happen with the autophagy deficient ones. So these cells, they have a problem in removing and clearing their cytosol from lipid droplets. And then the idea was to understand if this has an impact in the fate of infection, in bacterial replication, right? So we did some quantification of the size of these lipid droplets, and even though we have more lipid droplets in the autophagy deficient cells, they are smaller, which is in line with the idea that autophagy targets smaller, the smaller lipid droplets, okay? So we saw the very same thing with ATG5 deficient cells. So the phenotype is not ULK1 dependent, but seems to be autophagy dependent, okay? So because we observed a strong association of bacteria and lipid droplets, so here we have, I don't know if you guys can appreciate from there, but we have this strong association of bacteria with lipid droplets, which follows the same kinetics of the accumulation of lipid droplets. So we thought that it could maybe we have a relation with bacterial replication. So we did. We know from the literature that autophagy deficient cells, they have a defect in clearing the cytosol from bacteria. We had this long time ago. We infected ATG5 deficient cells and ULK1 deficient cells, and we observed that the cells they have defects in controlling bacterial replication, and we thought to understand if this could be linked to the accumulation of lipid droplets that we observe in the autophagy deficient cells. So because we know autophagy is important for the degradation of bacteria, right? I think we thought that using autophagy deficient cells to demonstrate that accumulate lipid droplets, to try to correlate that with the accumulation, the higher number of bacteria with the higher number of lipid droplets, it wouldn't work, right? Because we know autophagy by itself, it's important for the degradation, the direct degradation of bacteria. So we used this approach in which we used lalistat. Lalistat is an inhibitor that blocks the lysosome acid lipase, right? So it blocks the degradation of lipid droplets. Okay, so the idea it was to induce, to block to block the degradation of lipid droplets, to induce their accumulation and look to bacterial replication. And so we can see that lalistat, this drug, induces the accumulation of lipid droplets, very similar to what we see with chloroquine. We see that wild-type cells treated with lalistat and infected with listeria, they have increased numbers of bacteria present in their cytosol. And when we infect wild-type and ULKO deficient cells in the presence or absence of lalistat, we see that wild-type cells treated with lalistat, they acquire this phenotype, very similar phenotype to the ULK1 deficient cells. So suggesting really that inducing the accumulation of lipid droplets in the wild-type cells make them make bacteria replicate better, right? The treatment of ULK1 deficient cells with lalistat has had no impact. Okay, so this was what we've been doing in the last, let's say, 10 months. I will stop here, and I would like to thank people from our lab, especially Thales, Jones, and Bia. I would like to thank Patricia and Vinicius for discussions and reagents. People from the group of Marcelo Boza. I would like to thank Cristiani for reagents, expertise, and very helpful discussions in lipid droplets. Heitor, and funding agencies, the Institute, and you for your presence. Thank you.
All right, Leonardo, thank you very much for your talk. It was very exciting. I would like to apologize both to Leonardo and the audience for all the troubles that we had. That it's not usually what happens, but it's not always that we have the pleasure of having the presenter here with us. So I guess we are a little bit with a little bit of rust to get the whole system set for the talks. But thank you, and the floor is open for questions from the students first. So if we have questions here, if we have questions in São Paulo from the students.
Por enquanto tá sem. É. Hey, Bruno.
All right.
Can
So
you guys hear me?
get the ball rolling. I'm going to ask myself a question. So you showed that inhibiting the degradation of lipid bodies or lipid droplets, as you prefer to call. Cristiani will have a talk with you afterwards. But if you inhibit degradation of lipid droplets, you actually increase the number of bacteria. It's the two parts of this question. is that these lipid droplets, they are being used to target bacteria for degradation. They are being the lack of lipid droplets that make these bacteria more susceptible for degradation. And if you do the experiment the other way around, if you increase lipid droplets by several means, like unsaturated fatty acids, they increase the lipid droplets, do the infection get controlled more easily?
So first, it's important to say that this observation is for Listeria, right? So we have work from Patricia Boza with E. coli showing the contrary, meaning that lipid droplets, they play an antibacterial role, right? So it's going to depend on the bacteria. The other question, we use lysostats to increase the number of lipid droplets. So because why not using oleic acid, for example, because I think I have no proof on that, but I think the lipid droplets that accumulate upon oleic acid simulation would not be the same as those that accumulate during infection, right? So lipid droplets are variable in size, in proteome, in their lipidome. So I think we tried some experiments at the beginning with oleic acid treatment, but we didn't see differences. The other question, if LDs target bacteria for degradation. This is a very nice question. 2 weeks ago, I was in the Congress of Microbiology, the Brazilian Congress of Microbiology. I was discussing with Jones through WhatsApp. And he had this idea that lipid droplets can work as a tag for bacteria. Because you have a huge number of bacteria that are associated with lipid droplets. And recently we had 2 papers on Nature or something like this, demonstrating that ATG14 is a receptor for lipid droplets. So ATG14 binds to lipid droplets and send lipid droplets for degradation. So we have this idea that we are trying to gather the tools to do a first experiment in which the idea is to check whether lipid droplets are tags for bacterial degradation, right? So I'm not sure if I answered everything.
Yes. Thank you. Thank you very much. Actually, we have a question from Miriam, if she comes up here to appear on the camera as well.
So while we wait for more questions also, Leo, I thought it was really nice you showed both basically this crosstalk between the bacteria and the cells through the lipid droplets that they surround each other. And you mentioned that bacteria, some at least, have also lipid droplets. Do you think that there could be some exchange or some time of, it's easier for the bacteria to hide from the immune system if it exchanges, for at least for those that are more critical, maybe like Listeria, and that could be a virulence factor? Or?
I think it's really possible that bacteria can somehow manipulate the formation or the recruitment of these lipid droplets to their surface. But at the same time, again, I think we need to be really careful because the lipid droplets that accumulate upon E. coli infection are different from those that accumulate during Listeria, and maybe bacteria can modulate, how can I say, not recruit, but remodel the lipidome of the lipid droplets according to their needs or virulence factors.
Right. The questions here. If okay. If you could write down in the chat, we can relay the question to Leonardo. But meanwhile, just really outside of the box here. But you mentioned that ubiquitination is a really important step to the response to bacterial infection. But how come it's not exploited by the bacteria to escape? It is. So
We have many bacteria. We have different bacteria that are able to block this ubiquitination step, right? So by different the expression of different virulence factors, they can block the addition of ubiquitin to their surfaces.
Right. Thank you. I don't know if we are going to get any more questions. Time.
Diego said that they had a question, but they can't get.
Okay. So I guess one question that has been one of the main interests in our lab is the role of these the lipid droplets in lipid-mediated generation. Do you have any evidence that these may also play a role, that by assembling these lipid droplets, you could actually have lipid mediators being generating and influencing how autophagy? That's one thing that I learned today. I always said autophagy, and I learned that autophagy, but that's okay.
I think the right version is autophagy, but
Oh, really?
I've been using autophagy for.
Okay, no, you're the experts. I'll follow you. But do you have any idea if lipid mediators derived from the lipid droplets will play a role?
So this is in the list of things we will look with the help of Cristiane, because there are many studies in the literature demonstrating that autophagy deficient cells, they produce more cytokines, inflammatory cytokines. Okay, so this is let's say pretty well established, and we think that this excess of pro-inflammatory cytokines may derive from the accumulation of lipid droplets, right? So we had an interesting result in the weekend, more or less going into this direction. But we are going to partner with Cristiane to look for lipid mediators to see whether they accumulate in autophagy deficient cells due to the accumulation of lipid droplets. I think it's very likely that we will have more lipid droplets in autophagy deficient cells because of the accumulation of lipid droplets.
All right. Thank you very much. I think we accumulated a lot of technical problems. Oh, Maria has a question. Let's see if she can.
Hi. Do you?
Oh, okay. So please, Maria, go ahead.
Thank you. Very nice talk, Leonardo. I was wondering about other pathogens, not bacteria, but trypanosoma, for example, which are intracellular, cytosolic, and they go on ubiquination, I think. But are they also Yeah, I mean, I never heard anything about autophagy of Trypanosoma cruzi. Is there anything done?
Yes, Darius Group in 2017, they show in a paper on JBC that, I don't remember if it was Leishmania or T. cruzi. Yeah, but for Leishmania, I'm pretty sure that they show TLR-dependent autophagy. But for T. cruzi, I really don't remember, Maria.
Okay. Thank you. Very nice talk. And I think
you.
you have the French accent for autophagy.
Yeah, maybe.
Bye-bye. Thank you.
All right, thank you very much. Yeah, I think you solved that. You solved that, Maria. Thank you. Any more questions?
So, Leo, do you think there's anything that should be worried about when you block autophagy that with mitochondria turnover? Could that be changing something about pathogen?
No, for sure. But that's why we use this approach to block only lipid droplets degradation, because I mean autophagy is so important as a basal mechanism for the maintenance of homeostasis that for sure it may have an impact.
All right. So if you have no further questions, I'd like to thank everybody for the audience today here in São Paulo, on YouTube, and everywhere else in the universe that has been watching Leonardo's talk. Thank you very much, Leonardo, for your talk. It was really nice. I also appreciate the effort that you put into bringing all the history of autophagy. I really learned a lot today. Thank you, and I hope to see everybody once again next week for another installment of the Immuno webinars. Right? Thank you very much. Bye bye.